So now complex tissues. Complex tissues means this tissues formed by different kinds of cells, so called complex tissues or heterogeneous tissues. Generally, complex tissues help in conduction, so complex tissues are also called vascular tissues. These vascular tissues first appear in pteridophytes, and plants with vascular tissues placed under tracheophyta. Tracheophyta includes pteridophytes, then gymnosperms and angiosperms. Under these primitive tracheophytes, pteridophytes, advanced tracheophytes, angiosperms. Rightly, these vascular tissues, it includes xylem and phloem. And these vascular tissues, two types, primary and secondary vascular tissues. Primary vascular tissues, these are primary xylem and primary phloem. These primary vascular tissues are developed from procambium. Then secondary vascular tissues, it includes secondary xylem and secondary phloem. These secondary vascular tissues are developed from vascular cambium. So point, vascular tissues, xylem and phloem, there are two types, primary and secondary vascular tissues. Primary vascular tissues, xylem and phloem, developed from procambium. Secondary vascular tissues, secondary xylem and phloem, developed from vascular cambium. Then the xylem and phloem terms were given by xylem and phloem terms were given by Nageli. Xylem is also called hydrome. Phloem is also called leptome or bast. Here, xylem and terms were given by Nageli. Hydrome and leptome terms were given by Haberland. Next, individual description. First one, xylem. Xylem help in conduction of water and minerals. Now, parts of xylem. Xylem shows trachary elements. Then Xylem Parenchyma and Xylem Fibers. These are parts of xylem, trachary elements, Xylem Parenchyma and Xylem Fibers. Rightly, conducting cells or conducting elements of xylem are called trachary elements. It includes tracheids and vessels. Tracheids and vessels. So total parts of xylem, tracheids, vessels, Xylem Parenchyma and Xylem Fibers. Complete xylem is dead tissue, but living tissue of xylem, Xylem Parenchyma. Then first one, tracheids, primitive conducting elements. Primitive conducting elements are called tracheids. Then tracheid is elongated tube-like cell. Tracheid, elongated tube-like cell, middle part is wide, then end walls narrow or tapering. In tracheid, that cell wall is made up of lignin. and a trachea is dead cell without protoplasm. So at center there is empty space. That empty space is called lumen. Generally, tracheids are partially overlapped, and in tracheids the conduction takes place through bordered pits. And tracheids present in all tracheophytes, means present in pteridophytes, gymnosperms, and also angiosperms. Main conducting cells in pteridophytes and gymnosperms, tracheids. In angiosperms, along with tracheids, that vessels present in angiosperms. Then next one, vessels. Vessels also called trachea. Vessels, another name for vessel is trachea. And what is vessel means? Vessels are many xylem elements or xylem cells. All these are placed one above the other and forms a row of cells. Then in end walls, perforations are formed. This end wall with the perforations are called perforation plates. Then point, there are two types of perforation plates. One is simple perforation plate, entire end wall as one perforation, or a compound or multiple perforation plate, end wall has many perforations. Then, like tracheid, even vessels are dead cells. When vessels dead cells, then without protoplasm, at center empty space is present, rightly called lumen. Because of this empty space, that a flow of water easily takes place from base to top or between vessel cells. So in both the tracheid and vessel, that secondary wall is made up of lignin. But this lignin deposition is of different types. One is this lignin deposition. It's called annular. Annular means that lignin deposition is ring-like. If lignin deposition ring-like, it is called annular. Then second one, that lignin deposition is a spiral. Spiral means the deposition is spring-like, called a spiral. Then next one is scalariform. Scalariform means that a lignin deposition is ladder-like. If lignin deposition is ladder-like, it is called scalariform. Then next one, reticulate. Reticulate means that lignin deposition is net-like. If lignin deposition net-like, it is called reticulate. Next one, this lignin deposition is in the form of pits. If lignin deposition is in the form of pits, it is called pitted type. These are types of lignin deposition present in both tracheids and vessels. Then here tracheids and vessels, they help in conduction, and their cell wall is made up of lignin. So along with conduction, they also provide mechanical support. Then vessels are characteristic feature of angiosperms. Vessels formed in angiosperms. But in some primitive angiosperms, vessels are absent. Vesselless angiospermic families, Winteraceae, Trochodendroaceae, and Tetracenteraceae. In these families, Tetracenteraceae center AC. In these families, the vessels are absent. Then vessels absent in gymnosperms, but some gymnosperms like cycas and netum, in that vessels are present. Not in all cycas species. There is one species, Cycas revoluta. In Cycas revoluta, that vessels are present. Generally, vessels absent in pteridophytes, but some pteridophytes like Selaginella and Equisitum, they contains vessels. So these are about tracheids and vessels. Next one, Xylem Parenchyma. The only one living tissue present in xylem is Xylem Parenchyma. Then the Xylem Parenchyma is a living cells made up of cellulose, and this parenchyma contains vacuolated cytoplasm, and there are two types of Xylem Parenchyma. One is axial parenchyma and second one is ray parenchyma. Axial parenchyma. This axial parenchyma arranged in vertical rows along with vessels, and generally primary xylem contains axial parenchyma. Then next one, ray parenchyma. Ray parenchyma means they are arranged in radial rows. Radial rows. And a ray parenchyma present in secondary xylem. Actually, function of Xylem Parenchyma, it help in storage. Like it stores starch and also fats. Along with storage, this parenchyma help in conduction. Particularly, ray parenchyma, it help in radial conduction. Radial conduction means that a lateral conduction. It is Xylem Parenchyma. Next one, Xylem Fibers. Xylem Fibers, they are sclerenchymatous. Xylem Fibers are sclerenchymatous. And Xylem Fiber is elongated cell. Middle part is wide. End walls narrow or closed. And Xylem Fibers, they provide only mechanical support. And Xylem Fibers are two types. One is libriform fibers made up of simple pits, and second one is fiber tracheids. These fiber tracheids made up of wadded pits. So these are parts of xylem, and main function of xylem is that conduction of water and minerals, that too only from base to top, that is called unidirectional, or such flow is called ascent of sap. Xylem is of two types based on origin. One is primary xylem. This primary xylem present from starting stage help in formation of primary parts, and primary xylem is developed from procambium. The next one, secondary xylem. Secondary xylem, also called wood. The secondary xylem is developed from vascular cambium. Developed from vascular cambium. And primary xylem is of two types. One is protoxylem, that is newly formed xylem called protoxylem. Generally, protoxylem contains narrow vessels. Then second one, metaxylem, later formed xylem, it's called a metaxylem. This metaxylem contains broad vessels. Then xylem differentiation. Xylem differentiation is of two types. One is centripetal and second one centrifugal. Centripetal differentiation Centripetal means xylem differentiation is from periphery to center, called centripetal. So in centripetal, protoxylem towards outside, metaxylem towards inside. Such condition is called exarch, and exarch is characteristic feature of roots. And second one, centrifugal differentiation. Centrifugal differentiation. In this type, xylem differentiation is from center to periphery. It means in this type, protoxylem inside, metaxylem outside. Such condition is called endarch. This endarch is characteristic feature of stems. In some primitive plants like ferns, both centripetal and centrifugal differentiation takes place. Because of centripetal and centrifugal differentiation, the protoxylem is surrounded by metaxylem. Such condition is called mesarch. So these are types of xylem. Next type of complex tissue is called a phloem. Phloem is also called leptome or bast. Then function of phloem, it help in translocation of food from source means photosynthetic part, to sink, that is sink means storage pods. Then parts of phloem, generally phloem shows sieve elements. Then it shows Phloem Parenchyma, Phloem Fibers, and companion cells. Point: conducting cells of phloem are called sieve elements. Then sieve elements are two types: sieve cells and sieve tubes. Complete phloem is living tissue. Dead tissue of phloem, Phloem fibers. And primitive conducting cells, sieve cells. Their structure resemble to tracheids. Then these sieve cells present in pteridophytes and gymnosperms. What is sieve cell? Sieve cell is elongated cell. Like tracheid, sieve cell is also elongated cell, and their end walls narrow or closed. Then lateral walls contains sieve pores, and an area with a sieve pores is called sieve area. Then sieve cells are living cells made up of cellulose, and they contains vacuolated cytoplasm. And a matured sieve cell is without nucleus. And these are main conducting cells in pteridophytes and gymnosperms. Next one, sieve tubes. Sieve tubes, structure of sieve tube similar to vessel. Just like vessel, all phloem elements, they placed one above the other. Then their end walls are joined. After that, in end walls, that perforations are formed. These perforations like sieve. Hence, these are called sieve plates. Generally, sieve plates are oblique, and the sieve plates are two types. Simple sieve plate, entire end wall is only one sieve plate, that is called simple sieve plate. Example for simple sieve plate, cucurbita. The next one, multiple or compound sieve plate. involve as many sieve plates called a multiple or compound sieve plate, and example for compound or multiple sieve plate is a whites. Then like a sieve cell, even sieve tube. The sieve tube is enucleated, matured sieve tube, enucleated, and it contains vacuolated cytoplasm. Then the cytoplasm shows streaming movements or that cytoplasmic movements. And the sieve tubes, they are useful for conduction, particularly the cytoplasmic streaming. They translocate food from one sieve tube to another sieve tube through this sieve areas. Even sieve tube wall is made up of cellulose. Then associated with the sieve tube, one or two or many specialized parenchymatous cells present. The specialized parenchymatous cells are called companion cells. Then sieve tubes and companion cells are connected by primary pit fields. Sieve tubes and these companion cells connected by primary pit fields. Then through these primary pit fields, the cytoplasmic strands are traversed. Then matured sieve tube is enucleated, but matured companion cell, it has nucleus, and the nucleus of this companion cell controls metabolic activities of the sieve tube. That is why sieve tube is enucleated, but it is living. And in sieve tube and companion cells present only in angiosperms. In gymnosperms and pteridophytes, these are absent. In pteridophytes and gymnosperms, these are sieve cells present, particularly in gymnosperms. Along with the sieve cell, that albuminous cell is present in gymnosperms. These are sieve cells and sieve tubes. The next one, Phloem Parenchyma. Like Xylem Parenchyma, even Phloem Parenchyma is of two types. One is axial parenchyma, arranged in vertical rows, present in primary phloem. Then secondary phloem. Secondary phloem contains that ray parenchyma. Ray parenchyma present in secondary phloem. This ray parenchyma, they help in lateral conduction. An important point is Phloem Parenchyma absent in monocot stems. Then last one, Phloem Fibers. Phloem Fibers, they are dead cells. Then sclerenchymatous. Dead cells, sclerenchymatous. Phloem Fibers are also called bast fibers. Then generally Phloem Fibers are absent in primary phloem, present only in secondary phloem. These Phloem Fibers are commercially valuable. Like that, some of the examples. This is a flax fiber, it is obtained from linum. Then hemp, it is obtained from that cannabis hemp. Then sun hemp, it is obtained from Crotalaria. Then like xylem, even phloem is of two types, primary phloem and secondary phloem. Primary phloem is formed from procambium. Secondary phloem, it is formed from vascular cambium. Then primary phloem is of two types, protophlloem, that is first formed phloem, metaphlloem. Metaphlloem is later formed phloem, rightly called metaphlloem. And this phloem shows only one type of differentiation. That is what means centripetal differentiation. So this is about phloem, and final conduction of phloem, and finally function of phloem is it help in conduction of food materials. Now next one is a small tabular form, parts of xylem and phloem present in which plants and absent in which plants we can see. Here pteridophytes, gymnosperms and angiosperms. These are tracheophytes. Then tracheids, vessels, sieve cells, then next one sieve tubes, the next one companion cells, and next one albuminous cells. So these are parts of xylem and phloem. So first one tracheids present in pteridophytes. Then tracheids present even in gymnosperms. Tracheids present even in angiosperms. It means these are the only conducting cells. Tracheids present in all tracheophytes or all vascular plants. Primitive conducting cells are tracheids. Then lone tracheids present in pteridophytes and gymnosperms. Vessels and tracheids present in angiosperms. Next, vessels absent in pteridophytes. Then vessels absent in gymnosperms, but vessels present in angiosperms. Next one, sieve cells present in pteridophytes. Sieve cells present in gymnosperms, but sieve cells absent in angiosperms. Sieve tubes absent in pteridophytes, absent in gymnosperms, but present in angiosperms. Companion cells absent in pteridophytes, then companion cells absent in gymnosperms, but companion cells present in angiosperms. Next one, albuminous cells, right? Albuminous cells absent in pteridophytes, present in gymnosperms, absent in angiosperms. So this is clear. So these are conducting cells of xylem and phloem. So one small primitive conducting cells in xylem, tracheids. Advanced conducting cells in xylem, vessels. Primitive conducting cells in phloem, sieve cells. Advanced conducting cells in phloem, sieve tubes. Rightly, sieve tube and a companion cell, both are developed from same mother cell. So sieve tube and companion cells are called sister cells. That is why if sieve tube is not formed, companion cell is also not formed. Your vascular cambium, it produces two types of wood. One is heartwood and second one is sapwood. Here vessels of heartwood is blocked by balloon-like structures. These balloon-like structures develop from Xylem Parenchyma called tylosis. That is why vessels of heartwood not useful for conduction, and sapwood, this newly formed wood, it is useful in conduction. Rightly, these tyloses are called Xylem plugs. Then next one, phloem. In phloem, vessels. In phloem, inside sieve tube, a special protein is present. This special protein is called P protein or slime. P protein or slime, they blocks these sieve plates. So conduction is stopped. Generally, it happened in injured phloem. Then old phloem. In old phloem, callous plugs are formed. Callous plugs means your adjacent sieve tubes connected by cytoplasmic strands. Around that cytoplasmic strands, a thin carbohydrate layer is present. This thin carbohydrate layer is called a callus. And old phloem, this callus blocks that sieve plates, so rightly called callus plugs. This is additional information for Xylem and phloem, because last year in AIMPMT, these Xylem plugs or tylosis question asked. Next type of permanent tissues, special tissues. Special tissues, also called secretory cells. They produce substance and release substance as a result of metabolism. The special tissues divided into two types. One is external secretory cavities and internal secretory cavities. External secretory cavities, it includes digestive glands, then nectar glands, then hydathode. Internal secretory cavities, they includes schizogenous cavities and lysogenous cavities. So first one, digestive glands. Rightly, digestive glands, characteristic feature of insectivorous plants or carnivorous plants. Insectivorous plants or carnivorous plants, They contains digestive glands. These digestive glands secrete proteolytic enzymes, means protein digestive enzymes like pepsin and trypsin. Then they digest that proteins into nitrogen. Then plants absorb that nitrogen. These are digestive glands. Second one, nectar glands. Nectar glands, characteristic feature of entomophilous plants or insectivorous plants. This nectar is commonly made up of sucrose, glucose or fructose. By this nectar, plants attract insects. This nectar is of two types. If nectar present on